It is my pleasure to introduce Dr. Roger Kamm this afternoon. Dr. Kamm joins us from MIT, where he acts as a Cecil and Ida Green Distinguished Professor for Biological and Mechanical Engineering. Since joining as a faculty member of MIT in 1978, Dr. Kamm has become a world-renowned expert in biomechanics, directing the NSF EDIS program that many of us are familiar with, chairing national and world councils on biomechanics, as well as co-founding AIM Biotech and Cardiovascular Technologies. He has been the recipient of multiple Lifetime Achievement Awards, and in 2010, he was elected to the National Academy of Medicine. His research currently focuses on developing vascularization models of healthy and diseased organs using microfluidic technologies. Today, he will be presenting the plenary lecture titled An In Vitro Neurovascular Model for Alzheimer's Disease.
Thanks so much for the invitation and also for the kind introduction. I'm not using the microphone right now, but I can. So if anybody can't hear me, just let me know and I'll switch over. Okay, everybody seem okay now? All right.
Yeah.
So it's nice to be here again. It's like a second home. I've been out here quite a bit before. Our three partners in our EBIC Center, our STC that Martha referred to, were Illinois, Georgia Tech, and MIT. So I made a lot of trips to Georgia Tech and a lot of trips here, and enjoy my time at both places. So what I was going to talk about today is some work that we've been doing. The work in our lab is mostly in two areas, all sort of in microphysiological systems, but focused either on metastatic cancer or more recently on neurological disease. So really what I'm going to talk about today, obviously, is the neurological disease work. And going back to about 2015, I think, is when we first got started in that area. So I've got to give you a little bit of background information first to sort of bring you up to speed. And then I'm going to give short segments on different projects that we've been working on using these MPS, microphysiological systems, for different applications. So first of all, I never know if I'm going to get to the end of my talk, so I always like to give my acknowledgements first, and especially the postdocs, researchers, graduate students who really have done all the work that I'm about to present. Actually, the names of some of them aren't here because some of them have gone on to bigger and better things, but I'll highlight them as I go along in terms of who contributed to the work at each time, in each case. Also have a number of collaborators and our funding mostly for this work comes from the NIH, NINDS, and the Cure Alzheimer's Fund. And we're also just getting started with a industry consortium that has either 4 or 5 companies, major pharma companies involved that are interested in the brain modeling that we're doing as a basis for developing models that they can use for drug development and drug screening. So with that as an introduction, I'd like to start with this slide, which sort of gives a broad spectrum of what I think of in terms of microphysiological systems. These, as I think most of you know, are essentially organ-like systems or tissue-like systems that are in microfluidics. And they can range anywhere from something that's more sort of engineered in the sense that the cells are seated onto surfaces in a
I'm sorry, the slides aren't advancing on the screen. So,
oh, that's
yeah,
interesting.
I've
I've never seen that before.
Yeah, and I have no idea how to fix this.
Oh,
Okay, anybody want to voice in suggestions? Okay,
try again.
That has happened to us before when we shared the entire display instead of the PowerPoint app. So I don't know if.
I'm displaying the PowerPoint app right now.
okay.
I wonder if it's because see, now I can't get out of this either.
Okay. Gotta love technology, right?
Okay, let me stop sharing. I'll start over again. Okay, so I will share the app. Try that again. At least it's the same one that shows up. I might just leave the stuff up in the window.
Okay.
Okay,
Okay,
so
that's-
that's not moving again.
No, maybe I just need to advance it while you advance things.
Can you do that?
No, I can't. Oh.
I could go all the way out of Zoom. Try that.
Um, Anybody run into this
Yeah,
having
it's a
success?
Mac issue. It's a,
Mac thing?
yeah, I have the same thing. I don't know how to fix it. Unless you don't go to presentation mode. But yeah, it's a Microsoft Mac issue.
Lovely.
You guys format, there's a way to just share the little window.
Okay, now this is working. It has
Okay.
the, I wonder if I can get rid of the pictures on the side.
Yeah, you should be able to minimize that.
Should be able to do this.
Okay.
Now, let's see.
It's currently synced.
I can't advance anything now.
Oh,
Maybe I need to go down there.
Click on it. Once you click on
There
it,
we go.
okay. And
And now it
it's,
should
it's
be
all
okay.
synced.
Okay. Let's see if it works.
Thank you. Sorry about that.
Okay. So as I was saying, we can go all the way from more engineered systems. This is the emulate microphysiological system that got a lot of press back when it came out in 2010. This is a BioBot, which was generated here at Illinois by Rashid Bashir and Tahir Saif and company. This is a neuromuscular junction that I'm going to talk a little bit about today, and then this is a brain organoid. And as we go from left to right, we become more and more reliant on the ability of the cells to self-assemble and gain functionality on their own, rather than trying to program or engineer it by placing the cells in certain locations. The disadvantage is that we don't have very much control over how the organoids grow. The advantage over here is that it's much more reliable. It can be, and we can even predict its function in a lot of cases, but it doesn't take advantage of the emergent properties or the self-assembly properties of the cells themselves. So what I'm going to talk about today are all these topics. Let's see if I can get to most of them. I'm going to, first of all, talk about a non-vascular model. Most of our work is in vascular systems, but the first one I want to talk about is a neuromuscular junction model, which is one that we developed through as part of EVIX, and that we used as a model for ALS. Then I'll talk a little bit about the vascularization models that we've developed, how we do that. One example of that is the blood-brain barrier that we've been using for a lot of applications, and which is really the main aspect of the industry consortium that we're starting up now. And then some applications of blood-brain barrier, cancer metastasis to the brain, looking at nanoparticle transport across the blood-brain barrier, and then finally adding neurons to the system. So we have really a miniature brain in this particular case, to look at Alzheimer's disease. So first of all, the neuromuscular junction model. And by the way, if anybody has questions during the course of the talk, feel free to just raise your hand. I'm happy to answer questions as we go along. This is something that I know June and Martha and others have seen before, but this is work that was done by Tetsuya Osaki and Sebastian Uzel, actually started by Sebastian, a graduate student of mine, then followed on Tetsuya as a postdoc. And basically the system is what you see here. The idea is if we wanted to develop a system that had neurons in one location, muscle in another, but the two are communicating, so that we could activate the neurons, and the neurons then separately would activate the muscle to contract. So we did that by using ES-derived neurons that we could insert in this region over here. These were separately differentiated over a period of about 13 days. And then at the same time, simultaneously, we were differentiating and first of all, inserting muscle cells over in this compartment that would then consolidate around these two posts into a muscle strip, similar to the biological robot or the biobot that I showed in the previous slide. And then as these neurites extended out, connected between the neuron bundle and the muscle bundle, which you see in real life here, we could activate the neurons. These are optogenetically modified, so they express channel rhodopsin, so we can shine light on those neurons and get the muscle to contract. And here what you see, this is the neuron cluster over here. This is at the end of the experiment after 14 days in co-culture between the two cell types. The neurons are primarily over here. The muscle is over here in red. And you can see the neurites actually extending out from the neuron cluster going into the muscle. And I'll demonstrate that it's actually activating the muscle. So here's a single slice of a confocal image. You can see better the individual neurites coming out, branching, and then connecting up with the different myotubes inside the muscle bundle. This is a collapsed Z-stack. We're looking at the whole collection of neurites here that enter into the muscle. And then this is just a blow up of this region right here. And the way we measure the force is the fact that the posts that are there are compliant. So as the muscle activates, the muscle contracts and the posts bend. And we can see here, this is one of the posts. This was a movie at one point, but I lost the video. So you can just imagine that these now, when the muscle contracts, as you can see here, this post moves a little bit. And because we know the stiffness of the post, we can back out the force that's being generated by the muscle strip itself. Okay, so we can use this as a platform then to evaluate muscle performance. And we can do it in various circumstances. So we're interested in looking at ALS. So we and some of the results are shown here. I'm skipping over a lot of the details here, but basically what we did is we developed those two models. And then we wanted to see whether we could use this as a basis for drug screening. And there happened to be two drugs at the time, at least. This is back in 2018. There are two drugs that were in either a phase 1 or phase 2 clinical trial, bosutinib and rapamycin, both of which act on increasing autophagy. So increasing the ability of the cell to get rid of garbage protein. And if we looked at using these drugs in our two models, so the gray bar is the healthy motor unit, the black bar is the ALS motor unit. And I should point out too that the muscle in both of these models is the same, okay? The muscle is IPS derived, the neurons are derived from either a healthy subject or an ALS patient, okay? So we're measuring here is the forces generated by the muscle. And this is on day 7 of co-culture. You can see some differences starting to emerge here, but by day 14, it's pretty clear that the healthy muscle is generating a much larger force than all these others over here. The ALS motor unit it's about a third to a quarter the amount of force that's being generated. But if we used either bosutinib or rapamycin or in combination, we could recover that force generation capacity a little bit. In other words, the force increased by about a factor of 2. So we could actually see the effect of the muscle or the effect of the drugs on the muscle. And keep in mind here that, again, the muscle is the same in each case. The only thing that's different is the neuron cluster. You can see also, if you look at these muscle clusters, there are some differences that you can observe and identify by eye, but most of it really has to come about as a result of the functional testing, the functional screens that we can do within our system. The other thing we could look at is cell death in the muscle again, because we're really interested in muscle wasting that occurs with ALS. And if we looked at, for example, this is the expression of caspase 3-7, where we have the lower levels correspond to low level of cell death, the higher levels correspond to high levels of cell death. So you can see in the ALS motor unit, in the ALS muscle, we're getting a lot of cell death occurring, about 3 times as much as in the healthy unit. And we could recover that either by rapamycin by itself, Or if we combine the two, we actually got some synergistic effects, so we could actually reduce it more. So we could show in this system that the combination therapy was beneficial over the single therapy in the two cases. Now, we did a little bit additional work looking at the mechanisms that were at play here. And this is work that was done by Tetsuya Osaki. There's a paper that's available online. We'd submitted to Neuron, went back and forth a couple of times in reviews. Eventually it was rejected, and Tetsuya had already gone over back to Japan. So as a result, paper's sitting there. Okay, he's now moved back to MIT in a different lab, but we're going to finish that up. But what he had done was to demonstrate that the effects that we just saw on the previous slide were due to the fact that the ALS motor unit can actually transfer these both inflammatory cytokines, this is membrane-bound TNF alpha, and TDP-43 aggregates. So TDP-43 is one of the known mutations that occurs in ALS. Turns out that what we thought were a sporadic case of ALS actually was a familial type, because it did have several mutations that are associated with ALS. But these aggregates of TDP-43 that originate from the neurons actually get transported along the neurites. So in a separate experiment, Tetsuya looked at, he could actually plate the neurites down on the surface so we could get better ability to track vesicles that are being transported along the neurite. And so we could look at both retrograde and antegrade transport and identify what the vesicles were containing as they were being transported. And what he showed was that there was enhanced transport of these vesicles that contained the TDP-43 from the motor unit, or from the motor neuron out into the muscle. Once they got to the axon, they could be released either as individual molecules, the TDP-43, but more likely, what we found is that there were vesicles that were being released at that motor unit that would then diffuse across, enter into the muscle, and then cause the wasting of the muscle that we saw. And that's consistent then with what we saw in terms of the drugs that we tested. We demonstrated that skeletal muscle pathologies could then be improved by increasing the autophagy in the ALS motor unit, which decreased both the accumulation of the TDP-43 aggregates both in the neurons and in the exosomes that are being released out here and entering into the muscle. So that's one story. What I'd like to do now is talk a little bit about the vascular models we developed, and then how we apply those vascular models to neurological disease. This is a summary of a lot of work over a period of probably about 10 years. The most recent work was done by one of my postdocs, Joe Ofedu. And what we demonstrated over that period was that we could grow these microvascular networks like you see here. This happens to be a collagen type 4 stain that's showing the basement membrane of the vascular networks. These are grown within a microfluidic system. But if you compared this to the morphology that you see in the brain, and you'd find that it was very similar. Okay, in terms of dimensions, the length, the diameters, and so forth. We could grow vascular networks like this from various types of endothelial cells. These are human umbilical vein endothelial cells, or iPS-derived cells, primary cells, or even patient-specific cancer cells. Typically in co-culture with various types of stromal cells. The stromal cells are kind of the feeder cells that help the vascular networks to form and then stabilize subsequently. So these are the different types of fibroblasts or stromal cells that we've used. In the blood-brain barrier model, we chose to use the pericytes and astrocytes, brain pericytes and astrocytes, both primary cell types. I also had these images up here on the right that I wanted to point out. We could also do electron microscopy within these systems, and they allow us to really look at the tight junction between the two cells in great detail, and also look at vesicular transport here with false colors, where we're looking at vesicles being transported across the endothelial barrier or vesicles that are still attached to the cell membrane. Okay, so there's a lot that we can do with this. And as I mentioned, much of our work had to do with cancer. So we use these vascular networks to look at transport of tumor cells into a microvascular network, and then the extravasation of those tumor cells out into the surrounding tissue to form a secondary tumor. This is the sort of a typical device that we use. So you can see there's a pink region in here where we can insert cells and gel. And then we've got two side channels, left and right here, where we can introduce media, we can control the pressures, we can control the concentration of different factors that, for example, if we want to set up a gradient of concentration gradient in the gel, we can do that easily. But if we insert into this gel region a combination of endothelial cells, any of the endothelial cells that I showed you before, and the fibroblasts that I showed you, over a period of time, these cells, which initially look like this, form into this three-dimensional network. So here's one of the three-dimensional networks that's formed, and here the green spheres are tumor cells that once the networks form, we just set up a pressure gradient between this side and that side. We flow the tumor cells in. Some of the tumor cells get stuck, they become activated, and then they transmigrate out into the surrounding tissue. So here, this is before the transmigration occurs. I'll show you a little bit more of that later on. Here are two tumor cells that are inside one of these capillaries. You can see we can visualize this at very high magnification. This is 6DX. And we can also flow through these networks. I mentioned that we can control the pressure drop across them. And under a flow experiment, here we're looking at interactions between two tumor cells that are stuck in these small capillaries. And the red cells here are neutrophils that are interacting with the tumor cells, and actually through the signaling between the two cell types, they enhance the ability of the tumor cells to escape. Okay, so we've looked at a number of different immune cells. I'm not going to go into that today, but we found that both the neutrophils and platelets at least, and probably monocytes as well, influence the ability of the tumor to form in the metastatic setting. So from there, let me take you to the blood-brain barrier. And just for those who maybe are not that familiar with it, the blood-brain barrier is the tightest vascular barrier in the body, and has good reason to be. Brain doesn't tolerate a lot of the substances that are normally found in blood that other organs need. So instead of allowing that transport to occur on a pericellular pathway between the cells, there are a lot of transporter proteins in the endothelial cells that line the blood-brain barrier. And you can see some of them here, things like MRP1, PGP, LRP1 is around here somewhere, where you can get active transport either through vesicles, you can get absorptive mediated transport where the molecule absorbs to these vesicles and then get actively transported through various mechanisms. The brain endothelium selectively then allows the passage of certain substances in the blood into the brain. There's a lot of interest on the part of pharmaceutical companies to understand this mechanism, these mechanisms, and how to take advantage of them, either to prevent the passage of stuff across the blood-brain barrier or to facilitate it. So for example, with neurological disease, Parkinson's, Alzheimer's, Huntington's, and so forth, you need to get drugs into the brain. And what you can do is you can hijack some of these mechanisms in order to use these active transport mechanisms for getting the stuff in. And I'll show you one example of that in a few minutes, where we develop some nanoparticles that do that. First of all, the blood-brain barrier itself, well, in this case, we developed it from three different cell types. These are human iPS-derived endothelial cells, primary human brain pericytes, and primary human astrocytes. Okay, just actually in the past 2 weeks, we demonstrated now that we can get all these cells from iPS cells. So now we can get the entire blood-brain barrier model from patient-specific cells. But at this time, material that we published in 2018, this is what we used. We mixed the three cell types together, and again, we took advantage of the self-assembly of the different cells. When we injected these cells into the gel region here, they self-assembled into this vascular network. Here, the green are the endothelial cells, and the other colors are the pericytes and the astrocytes that are mixed in around the surrounding region. Those three cell types, by the way, were selected not just arbitrarily, but they're the three cell types that seem to be instrumental in terms of establishing the barrier function of the blood-brain barrier. And then what we do to measure the barrier function is we make a measure of what's called the permeability. And the permeability is just a measure of the leakiness of the barrier. And we often look at it as a function of the hydrodynamic radius or molecular weight of the molecule that we're interested in transporting. So small molecular weight molecules get across pretty easily, high permeability. As you go up in molecular weight or hydrodynamic radius, it's more and more difficult for these molecules to get across. That's where they need to be augmented. I show this graph in particular because it's taken from not one of our publications, but one from Kamm Leong at Columbia. The gray region corresponds to rat measurements in vivo. The blue squares correspond to the measurements that are made from our system and that we published in our 2018 paper. So you can see we can recapitulate pretty well the permeability that we see, at least of these different size dextran molecules that are fluorescent so that we can measure their transport. We're in the process of looking also to see whether the system expresses all the important transporter proteins, but we're still in the process of doing that. I'll show you a little bit in just a minute. So what we found from this was that the barrier function improves as we add the pericytes and astrocytes. I haven't shown you these data, but if we measure the permeability with just the endothelial cells by themselves or either one of these by themselves, we get higher levels of permeability. So in other words, it's the three cell types together that give rise to these low barrier functions. And secondly, that these permeabilities are comparable to recent measurements in vivo. So we got a model that basically works. It also looks pretty good at the ultrastructural level. So here we're looking at, these are electron micrographs of cross sections of one capillary. And if you look closely here, you can actually see the remnants of what was a cell-cell junction. This is becoming a seamless barrier, which is sometimes found in the brain, which actually means that the cells are trying to prevent as much pericellular transport as possible. And here you can see the association between the capillary, the endothelial cell, which is false colored green here, and either an astrocyte end foot. Okay, the astrocytes, they send out these projections and they have these end feet. The end feet interact with the endothelial cells and the pericytes and help to maintain barrier function. And here we had one image where we had all three cell types in one picture: the endothelial cells, the pericyte, and the astrocyte end foot. We could also look at using immunohistochemistry. We can look at the expression levels of these different junctional proteins. These are the junctional proteins that hold the endothelial cells together. And we can also look at a couple of basement membrane proteins. Here we're looking at laminin and collagen type 4. And as you go from the single cell, just the iPS endothelial cells by themselves, to the co-culture with the pericytes, to the triculture with the astrocytes, you can see you get progressive increased expression levels of each of these different proteins or basement membrane proteins. So what we found was that we were getting in vivo-like morphology. These could be created from primary or pluripotent cells. They match known in vivo permeability values pretty well. I'll talk about the phenotypic data in just a minute. And all this can be done with a single injection in what's now a commercial chip. So looking at the transcriptomics, we're interested in looking specifically at these junctional proteins here. That's the first 7 rows, and also the various transporter proteins. So I mentioned LRP1. I'll come back to that later on. Here's GLUT1, transferrin receptor. And what I want to do is I want to draw your attention, first of all, to these two columns. These are the primary cells. First of all, the first column corresponds to the primary cells that are plated on 2D. And the column over here on the right are the same cells, but they're in triculture with the pericytes and astrocytes, but now cultured in a 3D culture. And what you'll notice is that there's a huge difference between the two. And most of the transcriptomic data that you see published, these are cells that are seated on plastic. It's going to be very different when you see them in 3D, and especially when you see them with the other cell types that are present in the tissue. And this, I think, is a nice way to demonstrate that. The other thing I wanted to point out is that if you look at the iPS-derived endothelial cells on 2D, they don't look too much different than from the primary brain-derived endothelial cells. But also they change quite a bit as you go from a 2D environment to a 3D environment with the triculture. And then finally, if you compare the primary brain endothelial cells in 3D culture to the iPS-derived endothelial cells in 3D culture, you see that there's a lot of similarities between the two in terms of the expression levels of both the junctional proteins that are upregulated in the brain and also these transporter proteins, suggesting that these iPS-derived ECs are very plastic in their phenotype. So if you co-culture them with the right cell types, they'll take on the phenotype of the brain endothelial cells quite readily, at least with the one iPS cell line that we used. We're trying others right now just to see if that's still the case. So now that we've got the blood-brain barrier model, you might say, well, what can we do with it? One of the first things we looked at was, due to our interest in metastasis, we thought we wanted to see whether we could gain some insight into why the brain is such a receptive metastatic site, despite the fact that it has such a tight vascular barrier. So we looked at metastasis to the brain, and we first of all developed the blood-brain barrier model in the way that I just described. And then once we had it, we could, this is just a typical image, but we could then observe the extravasation rates, the rates at which these green tumor cells escape from the intravascular space to the extravascular space. Okay, and that would allow us to calculate the percent extravasation of these, in this case, the MDA-MB231 cell. It's a triple negative breast cancer cell line, but we also then did it for another breast cancer cell line and lung cancer and prostate carcinoma, and showed that in each case, as you went from just the endothelial cells by themselves to the co-culture to the triculture, you got a progressive increase in the extravasation rates, despite the fact that the permeability is going down. And we would have thought, and we had thought up to this point, that the tighter the barrier, the less extravasation you're going to get. So if you want to reduce metastatic spread of cancer, just get those endothelial cells to form really tight junctions. Obviously, that's not the whole story. You really wouldn't expect it to be. So there's something else going on here. So wanted to try to address that question. So what we did is we turned to this cell line that was derived by the Maskey lab in 2009. And what these are the same MDA-MB231 cells. But now what he's done is he's passaged these through a mouse a number of times. So he injects them into intracardial injection, they metastasize to either the brain or the lung, and then he collects them from those organs. And he does this several times. So he gets here a collection of cells. These are the parental cells, the ones that he started with. The LM are the cells that preferentially metastasize to the lung. And then the BRM are the cells that preferentially metastasize to the brain. So we took those three cell lines, we got those from his lab. And sure enough, we found that if we did just the iPS-derived endothelial cells by themselves, the ones that don't really know where they are, okay, they haven't seen any brain cells yet. Both of these, the lung and brain, extravasate more highly, a higher percentage, but they don't. It's not differentiated between the lung preferential cells or the brain preferential cells. Only when you add the brain-like cells, here the pericytes, or here the combination of pericytes and astrocytes, do you see that there's a preferential a higher extravasation rate of these brain metastatic cells relative to either of these other two populations. Okay, so we were capturing some of the aspects of what Massey had seen in the mouse. We first looked to see whether, well, maybe it's the presence of the cancer cells, of the MDA cells, that is giving rise to this difference, but it wasn't, because here what we found is that when we added the MDA 231s or conditioned media from the 231s, In any of these systems by themselves, there was really no significant difference between the permeabilities that we measured. Okay, so that wasn't explaining it. So there was some evidence in the literature that suggested that, let's see, check it out. Okay, it suggested that the astrocytes might be instrumental in this preferential metastasis to the brain. So we looked specifically at what the astrocytes were secreting when they were co-cultured with the tumor cells. And this is all normalized to the chemokine or cytokine release profiles for just the endothelial cells by themselves with the MDAs. This is the pericytes plus the MDAs, this is the astrocytes plus the MDA, and we focused on CCL2 because we had a suspicion it was the astrocytes that were being contributing the major factor. You can see the whole cytokine array down here, and you can see the astrocyte plus MDA, this is the CCL2, and you can see that the highest levels of expression was when we had the co-culture with the astrocytes. So we did some experiments to see whether that was in fact not the case. And the first thing we did is we took recombinant CCL2, starting with our IPS system endothelial cells by themselves. We added the CCL2 to the system at different concentrations. And sure enough, we found that it gave rise to a progressive increase in the extravasation rate. We did the same thing in the IPS endothelial cell plus pericyte system. And again, we saw a significant increase in the extravasation rates when CCL2 was around. And then we did the opposite. We took the triculture system where the astrocytes are already secreting the CCL2, and then we blocked it, we inhibited it. And as we inhibited the CCL2, we found that there was a progressive reduction in the extravasation rate. Okay, all consistent with the idea that the CCL2 is a key factor in increasing metastasis to the brain. We did one more experiment. We took, let's get these out. There in the initial Massey paper where they had isolated these different cell populations, they had identified this other molecule, ST6 GalNAc5, as being instrumental in giving rise to the difference in metastatic rates. And we had found something different. So we wanted to see, are our results consistent with theirs, and can we observe the same effects that they saw with the ST6? So we took initially our parental line. This is and we normalized everything to that. This is a full change in a percentage in extravasation over a 24-hour period. In going from the parental to the brain metastatic cells, we saw the increase. That was just what I showed before. But then as these were knockdowns of either the FB6 or the CCR2. CCR2, by the way, is the receptor for CCL2. It's easier to knock down because it's knocking these down in the tumor cell, not in the astrocytes. If we knock down the ST6, we saw a significant reduction in extravasation. However, if we knock down just the CCR2, we saw an even more significant, larger reduction. And if we combine the two, knock down both the ST6 and the CCR2, you can see we've got the lowest level of extravasation. So we're able to not just confirm it was some previous results that have been demonstrated in mice, but extend it and show that there is not just one factor, but a couple of factors, and that they act in synergy with one another. We went back and we checked this in mice. These were experiments that were done in collaboration with the Taylor Jorgensen lab at MIT. So they did intracardiac injection of these cells, waited 24 or 48 hours, dissected out the brain, and then used confocal imaging to identify where the cells had metastasized. And we saw the same sort of thing. With the control, this is shRNA knockdown for CCR2, we got a significant reduction in the percentage of cells that extravasated into the brain tissue, both at 24 hours and even more so at 48 hours. Turns out the process of extravasation takes a little bit longer in vivo than it does in vitro, although our current models are actually taking longer and longer, suggesting that, well, I'd like to think that's because we're getting more and more in vivo-like, but there are a lot of reasons why that could happen. Okay, so the next thing I want to talk about is the work on the nanoparticle transport to the brain. And this was work that was done in collaboration with Paula Hammond at MIT, where she develops these neat particles. These are developed by layer by layer deposition that are functionalized. Ultimately, these are liposomes that are functionalized with angioPep2. Or actually, they can be functionalized with a number of different peptides. The one that I'm going to focus on is angioPep2. So this is a model where it's a xenograft cell line, GBM22, that metastasizes, that grows. It's a glioblastoma, that a brain cancer that's native to the brain. The spheroid is injected in the system. Here, all we do is we mix the blood-brain barrier cells with the tumor spheroid. We inject everything at once, okay? And then the blood-brain barrier forms, and then we can introduce the nanoparticles to the system. So the spheroid and the cells are injected on day 0, and then we do all the perfusion experiments on day 7. This is the apparatus that we use. This is the device. Again, it's essentially the same device that I was showing earlier. All the action takes place right in here, and here is one image. These are the glioblastoma cells in green, and here's the blood-brain barrier, the vascular network, shown in red. And this paper was just published this summer. So I mentioned there were a couple of different types of nanoparticles functionalized with different molecules. These are just showing that all the molecules, all the nanoparticles are getting into the system, and they get up close to the glioblastoma tumor. We identified, though, that the LRP1 is more highly expressed close to the tumor as compared to either if there's no glioblastoma tumor at all, or if we look in the same device, but far from the tumor. So that was what motivated us to use angioPep2 because it's the angioPep2 that binds to LRP1, which is one of the transporter proteins, to augment the transport into the tissue. So the bottom line was when these nanoparticles were loaded with cisplatin, which is a chemotherapeutic for cancer, that we found that there were higher levels of cell death in the case of the angioPep2 functionalized nanoparticles relative to either the bare nanoparticles or the free cisplatin, or no treatment at all, obviously. And that this is the death rate in the tumor, and this is far from the tumor, and this is close to the tumor. So we can see that the increased killing rate really occurred and it was relatively confined to the region of the tumor itself. So finally, I want to talk a little bit about the model we developed for Alzheimer's disease and cerebral amyloid angiopathy. What that is there's a tendency early on in the development, the progression of AD, where the A-beta, the amyloid beta protein tends to accumulate around the small vessels, and it's known to increase the permeability of those vessels. So we wanted to see whether we could demonstrate both the change in permeability, which is thought to be contributing then to the progression of the formation of the disease itself. And also if we could identify then that the A-beta plaques were growing around those small vessels. So we used a slightly modified device. Here, this is now the region where we inject the gel. There's also a central port that we're going to introduce neurons. And the neurons are either a wild type neuron or an Alzheimer's neuron. These are neural precursor cells. They're human, and they've been engineered. Either there's the non-Alzheimer's disease cells, and we're comparing these to different levels of expression of A-beta that's been done by genetic engineering of the cells. So the neurons are now in this is what you see up here, this semicircular region. That's this region in the center. The blood-brain barrier is all the way around. This work, by the way, is done by a really great postdoc, Jorgis Pavlo, and also with help from Claire Coe and others in the lab. So here, there's 2 injections, one injection for the blood-brain barrier, one injection for the neurons. And here the neurons are just in suspension. They're suspended in matrigel. They get put into the central region. And after 7 days of co-culture, you can see the blood-brain barrier. Well, you can see the blood-brain barrier in both cases. Here you can see that it's perfusable because we perfuse through a 40 kilodalton fluorescent dextran that helps us to visualize where the vascular networks are. However, we found that we had to go out long-term for these experiments. These are 30-day experiments now with the co-culture of the blood-brain barrier cells. So those 3 cell types, and then plus the neurons, in order to really demonstrate the Alzheimer's disease state. In order to do that, we needed to perfuse the system. So we designed a small pump. It's a very simple pump. You can see it here. It simply sits on top of it's a PDMS pump, sits on top of the region that has the neural system in it. There's one tube that comes here that just provides a pulse of air. And that pump will last for, if we're lucky, it'll last for 30 days. We have bugs in the pump that we're still working out, but for the most part, things generally work. So this is the course of the experiment. During the first 8 days, we're differentiating the cells. And 2D, we see the blood-brain barrier and the neurons in day 0. We connect the pump on day 7, and then we let it go for a while up to day 30, and then we look at the distribution. We use RT-PCR to look at both the profile of the cells, but also we immunostain, and this is mostly the data that I'm going to show, immunostaining for A beta and the blood-brain barrier, the tight junctions. So this is what the model looks like. And if you look closely, you can see the microspheres that are flowing through. They're blue, just to demonstrate that, yes, we are able to perfuse through the blood-brain barrier still. Not many of the cells actually get, not many of the vascular cells get into the central region because they don't like the matrigel as well. We're working on that right now. We've got some methods actually to perfuse into, we can perfuse into a liver organoid. We haven't tried perfusing into a brain model yet. But this is what the networks look like after they've been perfused for 30 days. So you can see the morphology has changed a little bit. Diameters have gotten narrower. Well, in general, they get wider, but there are fewer vessels around. So you can see this is a 200 micron scale bar down here. Most of these vessels are in the upper end of the range of what we saw when we first seeded the cells. But you can see from the dextran, the blue dextran, that really pretty much all the vessels are being perfused here. And this is just, these are some images here. This is with the red dextran, just showing what the vascular networks look like. And if we wait then 30 days, first of all, if we sample the media at day 7, we're sampling the media from the neuron culture portion, from the center part. And at that point, we can see, first of all, if we measure the permeability, we get not significant difference, maybe a little bit lower in the Alzheimer's model than the wild type. However, if we wait the 30 days, you can see the permeability now is about twice as high for the Alzheimer's cells as it is for the wild type. We could also collect the media from that central port, as I mentioned, and then we can measure the concentrations. Here we plot the concentration of A beta 38, A beta 40, and A beta 42. It's the A beta 42 that's thought to be associated primarily with Alzheimer's, but we find that all of these are highly upregulated, more highly expressed in the different models that we've been developing, the Alzheimer's model relative to the control model. You can see there's a break in the scale. So these levels are down around 50 and they jump up to the thousands. Okay, so there's a huge increase in the amount of A beta that's present. So, and by day 30, there's an interesting observation here that if you look at day 7, there's a high increase in the A beta 42, but then it comes down quite a bit. And we think what that is caused by is the fact that the A beta 42 is probably being sequestered from the solution into the plaques that are in the process of forming. So then we look for the plaques. And so here we stained for, this is staining for all A beta. We couldn't selectively stain for A beta 42. So the green is the blood-brain barrier, the purple is the A beta. What you can see is it looks like initially when we did this, we couldn't tell whether it was inside or outside of the vessels, just from the immunofluorescence. Although you can see there's certainly a lot associated with the vessels. So we went back and we did some further experiments where we could actually label the A beta. And we saw that sure enough, there is a cuffing, A beta cuffing that goes around the individual vessels that you can see here. So the cuffing is occurring and it's associated at least with the permeability changes that we observed. And this is the most recent work, and I wanted to make sure I showed this because Claire is an alumnus of University of Illinois. She worked in Jim Cummings' lab, and she's now a postdoc with me. And what she's been doing is doing essentially the same kind of experiment, but instead of seeding the cells individually in that central compartment, she's forming neurospheres and then putting collections of neurospheres into that central port so that we can get a higher cell concentration. The idea here is that we're hoping that the effects of Alzheimer's will show up earlier than the 30-day time point that we're currently looking at. And the initial evidence looks good. So what she's done here, you can see a collection of neurospheres that are inserted into the central port. The outside here, these are what are going to become the blood-brain barrier. These are just the injected cells initially. This is the schematic of the device again. And what she has found so far, and this is just a protocol that she uses for the experiment. Here are the neurospheres, and then there's a matrigel that's put on top. She's looked so far at the morphological characteristics of the neurites. She finds that by day 7, she could already see. Whoops, my laser pointer just died. By day 7, she could already see differences in the neurites being extended from the healthy neurons relative to the AD neurons. We are not seeing significant differences in the permeability as yet, and we haven't done the staining for A beta yet. These are hot off the press. I first got these results, I guess it was 2 weeks ago. So this is the direction that we're headed in. We're looking to see whether we can develop a completely now iPS-derived model of Alzheimer's. This is now still with the primary cells. But I mentioned that we have now evidence that the iPS cells are also working. It took a while to get that optimized. And in fact, not all iPS cells are alike in the sense of we've gotten a number of different iPS cell lines that are supposedly endothelial cells, some of which form vascular networks and some of which don't. So I think oops, should be my last slide. And I can't get to the last slide anyway. It's not letting me. The last slide. Okay. Anyway, the last slide is a summary of everything that I just presented. So I think we'll just stop there, and I'd be happy to answer questions if there's time. Martha.
Fantastically interesting, Roger. And wow, you have covered a lot of ground since I last heard you give a talk.
We've been busy. Yeah.
Do you have any speculation on how a beta 42 increases permeability? If I'm interpreting what you said, right?
Our guess, and it is just a guess at this point, is that it affects probably the junctional proteins. How it does so, we don't know. And I haven't seen anything in the literature yet to really point us toward one particular pathway. It seems as though it needs to form. Well, I take that back. It seems as though it's possible to change permeability even when it's in the soluble form. So these could be oligomers before we actually form plaques, or it could be the single molecules. So it probably isn't essential that they form cuffs. But more than that, I can't really say.
Interesting. Thanks.
Yeah. Yes.
The one now for the pump system, how is that recirculation work now in the market?
So it's a very simple system. It has there's a pumping chamber, and then there are two essentially check valves, one on the upstream side and one on the downstream side. And as we pump air into that chamber, it's a diaphragm, pushes down on the diaphragm, goes out through the upstream side, passes the diode into a capacitor. And then when we release the air pressure, it draws air in from another capacitor. So we're maintaining a fairly constant pressure drop across the vasculature. There's small fluctuations, but we can model all that. So we can calculate and redesign the pump depending on what the parameters are for the stiffness of the membrane and the pressure that we apply. Yes, please.
In your neuromuscular model, are you or anyone else studying like peripheral neuropathy of any sort, like the hereditary peripheral neuropathies that are primarily motor, like Charcot-Marie tooth disease, that kind of stuff?
We are not. We've been approached by a couple of different groups to look at, to use the model for different purposes. And to be honest, since I don't have work going on in that area in my lab right now, I basically have had to tell them, I'm happy to help you with the neuromuscular junction model, but we can't do experiments in our lab right now because we don't have the funding for it. But yeah, that would be an excellent application area.
That system, the same one, we have the image there for, are the compartments completely separate between the neurons and the full? I mean, probably not completely separate, but I heard you say you get the media just out of that neuron area, right?
Yeah,
How are you?
yeah, so the media, we use neuron media in the central port, and we perfuse vascular media through the others. In fact, turns out the neurons are relatively happy with a mixture of 50-50 of the vascular medium and the neuron medium. With the neuromuscular junction model, again, if you remember that, I was going to go back to that. There are two different wells for media. And what we do is we put neuron media in one side. Whoops. We put neuron media in one side. Yeah, you can't. So there's yeah, here you go. So these are the two wells that we have. We put the neuron medium over here. We put the media that the muscle likes over here. So the two diffuse relative to one another. So there's kind of a mixture, but it works. So, the media incompatibility is it's always an issue, but we found ways of getting around it. And we found that the cells tend to be a little flexible.
So when you extract the media, it is safe to assume that most of the stuff that you find in the media comes from one cell type.
So in this experiment, we haven't been analyzing the media. We've been using this mainly to look at the interactions between the neurons and the muscle, and mostly looking at what's happening in the neurites. With the other system, if we sample from the central port, that's sort of like the interstitial space in the brain. Okay, it's separated from the vascular media because the endothelial media is inside the vessels. There is some transport that occurs because a lot of the stuff that's in the media that perfuses through the vessels diffuses out through the wall. But it's a fairly well separated system. Yeah.
We have a question in chat from Andrew Ding, who says, I was wondering whether you have considered using transcriptomic analysis to examine how amyloid beta might be affecting permeability in the engineered Alzheimer models.
It's on our list. I mentioned that we do have additional funds coming in from this industry consortium. And one of the items that we've identified that we're going to do with those funds is to start to do a more detailed analysis of the transcriptome. We will probably need to do single cell analysis because we've got all these different cell types present. But obviously the techniques and the technology to do that is readily available as long as you have the money. The last that I heard is that it costs about $10,000 to do a complete single cell seq analysis from an experiment. So we're working up to that. Definitely something we want to do. Okay, thank you very much.